Okay, hello, welcome back to our biotech class. Then today we are going to study about 10th class biology, that is sunlight is necessary for photosynthesis. Sunlight is necessary for photosynthesis. So in the last class we stopped at the how hydrilla plants can produce the oxygen in the presence of sunlight that we discussed. Then in the continuation class we are going to study hydrilla plants can produce the oxygen in the presence of sunlight. Suppose John Hingenhaus he conducted some experiments that he took some hydrilla plants and kept in the water, and this hydrilla plants and water it is exposed to the sunlight. When it is done, there are some air bubbles are coming out through the water. That shows about these air bubbles are producing some gas. That gas is nothing but it is tested as a oxygen. Okay, that was the last class we conducted, and we explained about hydrilla plants can produce the oxygen in the presence of sunlight. Then here we are going to prove that how sunlight is necessary for the photosynthesis. So for this we need to have some material, that is what one potted plant. We should take one potted plant. Then this potted plant should be kept in the dark room for few days, 4-5 days. Take a potted plant, and this potted plant should be kept in the dark room for few days. What is the reason? Because of to stop the process of photosynthesis, or this plant is to be free from the photosynthesis, or this plant is to be free from the starch. That's why we selected one potted plant and kept in the dark room for few days, so that if there is any reserved food in these few days, that plant can be utilized for its survivalness and metabolic activities. So when the plant is kept in the dark room, there is no sunlight availability, so that photosynthesis process would be stopped. This we are going to prove practically through this activity. So here after few days, suppose taken one plant, we have taken one plant and it is kept in the dark room for few days. So within these few days this plant is free from the starch. Then after few days, take out the plant and keep it in the presence of sunlight and arrange one light screen onto the leaf. See, arrange one light screen onto the leaf. So we have arranged one light screen here, and this light screen has a cut design. Suppose example here, this cut design, there is a cut design which is S-shaped cut design. So except this S-shaped cut design, then this remaining part of the leaf which does not get sunlight, this remaining part of the leaf, this remaining part of the leaf which does not get sunlight, because light screen is arranged here. Light screen is nothing but a dark sheet of paper, just like a dark sheet of paper or a dark sheet is covered on the leaf, but this dark sheet has a cut design, that is S-shaped cut design. So through this S-shaped cut design only light is absorbed into the leaf, but remaining part of the leaf where we have covered the leaf with cut design or black sheet or light screen, through that place sunlight is cannot be enters. So after few hours you can observe, after few hours this plant and this total experiment, this total arrangement should be kept in the presence of sunlight in the for few hours. Then after few hours you can detach the leaf. So this leaf you can detach from the plant, okay, and spread it on the petri dish and observe with the help of iodine solution. Now you can pour the iodine solution onto the leaf. Okay, suppose if you pour the iodine solution onto the leaf, see the leaf, ఎక్కడైతే లీఫ్ కి మనం బ్లాక్ షీట్ ని లేదా లైట్ స్క్రీన్ ని కవర్ చేసినామో, ఆ లైట్ స్క్రీన్ ని కవర్ చేసిన భాగంలో గనుక ఒకవేళ అయోడిన్ వేస్తే, suppose if you pour the iodine solution on leaf which is covered with the light screen, this cannot be turned into blue or black in color. But where we pour the iodine solution onto the leaf which has the cut design is S-shaped, సో ఈ ఎస్ షేప్లో ఉన్నటువంటి కట్ డిజైన్ పైన కనుక మనం ఐడిన్ సొల్యూషన్ వేస్తే then it will turn into blue or black in color. So that shows about here, because of the cut design, here light is entered, sunlight is entered into the leaf, so that only that S-shaped part of the leaf is turned into blue or black in color. That shows about starch is formed or carbohydrates are formed in this S-shaped part of the leaf, but remaining part of the leaf which is covered with the light screen, which cannot be turned into blue or black in color. That shows about this part of the leaf has not received any sunlight. Because of only lack of the sunlight, this place is not prepared the food materials. That's why it is not turned into blue or black in color when we poured the iodine solution on it. So with this we have practically proved that sunlight is necessary for the photosynthesis. Just imagine, suppose water is absorbed through the underground, whereas chlorophyll is present in the leaf, and carbon dioxide is diffused into the leaf through the stomata. Everything is available to the leaf, but only due to the lack of sunlight energy, here carbohydrates are not formed where the leaf is covered with the light screen. So with this we understood sunlight is necessary for the photosynthesis. So this is about the sunlight is necessary for photosynthesis. You can next chlorophyll and another important thing, chlorophyll and photosynthesis. The last one is chlorophyll and photosynthesis. Let us see chlorophyll and photosynthesis. Chlorophyll and photosynthesis. So what is the relationship in between chlorophyll and photosynthesis? Very easy. See, chlorophyll is one of the most important pigment which is present in the leaves. So because of chlorophyll, the plants are green in color. They could carry out the photosynthesis process and prepare the food material by themselves. So that we have been studied several times in our lower classes. But here practically we are going to observe the relationship in between the chlorophyll and photosynthesis. So here John Ingenhouse, one of the scientists who says that John Ingenhouse, see, according to the John Ingenhouse, see what he says, green parts of the plant could carry out photosynthesis. Very most important quotation. Green parts of the plant could carry out photosynthesis అని చెప్తాడు అన్నమాట. Okay, green parts of the plant could carry out the photosynthesis. Then where is the green parts of the plant? Suppose leaves are green in color, so even some stems are also green in color. So like this, green parts of the plant could carry out the photosynthesis. This statement is given by the John Ingenhouse. So whenever he has given this statement about green parts of the plant could carry out the photosynthesis, then some other scientists, they have raised some questions against the John Ingenhouse. See, suppose what about the leaves which are red in color, or what about the leaves which are yellow in color, what about the leaves which are some other colors? Could they prepare the food material by themselves, or could they carry out the process of photosynthesis? So they have, other scientists, they have raised some questions against the John Ingenhouse, but he could not answer. Only he has said that green parts of the plant could carry out the photosynthesis process. But after several years, in the year of 1817, here Pelletier and Caventos అనే ఇద్దరు సైంటిస్టులు, in the year of 1817, Pelletier and Caventos, see, these two scientists, they extracted a green pigment from the, extracted a green substance from the leaf. They extracted a green substance from leaf. So this green substance they named as chlorophyll. Chlorophyll, very most important. In the year of 1817, Pelletier and Caventou extracted a green substance from the leaf, and this green substance is named as chlorophyll. Chlorophyll means here green leaf. in the textbook it is written in the bracket, chlorophyll means a green colored leaf. See, జాన్ ఇంజన్ హౌస్ ఇచ్చిన స్టేట్మెంట్కి ఇక్కడ చాలామంది క్వశ్చన్స్ అడిగేసరికి ఆయనకి ఏం చేయాలో అర్థం కాల. ఇక్కడ జాన్ ఇంజన్ హౌస్కి రెడ్ కలర్డ్, yellow కలర్డ్ అండ్ అదర్ కలర్ లీవ్స్ కుడ్ దే క్యారీ అవుట్ ద photosynthesis ప్రాసెస్ అని అడిగేసరికి సో he could not answer. But after several years in the Pelletier and Cavendish, they have experimented and extracted a green color substance from the leaf and they were named as it is a chlorophyll. See, they were named as it is chlorophyll. Chlorophyll means green color leaf. So this chlorophyll, when it is injected in the leaf, it could carry out the photosynthesis. But suppose if the chlorophyll is extracted from the leaf, if it is kept in the test tube and if we add water and carbon dioxide and sunlight, no photosynthesis can be taken place. But only the chlorophyll, when it is injected to the leaf, it can be used as a photosynthetic pigment and it can be used as a factor to prepare the food material by itself. But if the chlorophyll is completely isolated from the leaf and if it is kept in a test tube and if we arrange the carbon dioxide, water, sunlight, everything factors which are needed for the photosynthesis are arranged in the artificial conditions or in a lab, photosynthesis cannot be occur. So that chlorophyll must be intact to the leaf to prepare the food material by itself. So that is the most important. And then what about the red colored and yellow colored and some other colored leaves? Could they prepare the food material by themselves? Some other scientists, they asked some question now. So for this they have also answered that not only chlorophyll and some other xanthophylls, okay, carotenoids, okay, phycobilins. So this type of pigments are also present in the leaves. So these carotenoids, phycobilins, xanthophylls also could prepare the food material by themselves. They have also chlorophylls, that chlorophyll can prepare the food material by themselves. That shows about green parts of the plants are not only carry out the photosynthesis but also with besides the chlorophyll there are some xanthophylls, phycobilins, carotenoids and some other pigments also could carry out the process of photosynthesis. So with this, the question what they have asked the scientist, that answer is given from the Pelletier and Cavendish. So finally, this is the relationship in between the chlorophyll and photosynthesis. Okay, thank you.